So today what we want to do is zoom way in. So last lecture was about all these different cortical areas and all the maps in V1. And so at that kind of scale, what we were looking at was there's the center of gaze, and there's the vertical line in the visual field, and that's the horizontal meridian, and that's the lower and the upper field. We're looking at kind of like a relatively macro level. So what we're doing today is we're taking just one little tiny little chunk of that, one little tiny chunk, and then just blowing that thing up massively into a little chunk that we took out of the cortex. So the map is still there. So if you look, if you look at this little chunk, it has various things in it. We'll talk about like blobs and etc., different layers. But the map is still there. So the map is still there, sort of going this direction and this direction across the cortex. So that's still the retinal map. And what we want to talk about today is how do we embed all these different streams of information? So we talked about starting in the retina. You start off with some photoreceptors, and you immediately kind of differentiate them into these multiple streams that are multiple parallel streams that are going into the system with information of different kinds at each point. So that's the focus for today. So we're just going to sort of massively zoom in on this little chunk of cortex there, and just look at how the streams are sort of running through it, and then also emphasizing this idea that the way this works is you make some streams, and then they go to an area and they get all mixed up, and then you get some more streams coming out, higher level streams, but you can't easily sort of trace your way through one stream. But at any point, if you look at the outputs, there'll be streams. So that's going to be the theme for the next month or so, to sort of go over these different systems and see how the streams kind of diverge and come back together and diverge and come back together. Okay, so that was our intro, and so we're going to start off with general scheme for cortical layers. So cortical layers, we talked about layer because we're going to go into detail about all these different layers. So when we talk about a general scheme for cortical layers, it's like the rule of Serino. It's not completely 100% true. It's kind of like 85% true, 90% true, but it gives you some general sort of framework to look at connections between cortical areas. And so if we start off with a quote-unquote lower, you'll see why I put the quotes there, because there's so much feedback that information is going both ways. So here's a lower cortical area, and we can basically sort of name the areas, name the layers in terms of their function. So if you take a little chunk of that cortex, like this, we've got these different layers. So starting at the top, there was layer 1. Now what is layer 1? So layer 1, we call layer 1 the original input layer. So there's not too many cells in layer 1, and so when you look at, if you look at a cortical section, you'll see, a bunch of cells in layers 2 to layer 6, but layer 1 is pretty much empty of cells. There's a lot of dendrites up in there and a lot of axons, but there's no actual cell bodies. But, why do we call it the original input? Because if you look at something like a turtle, so if we look at a turtle, if you look at the turtle cortex, there's a layer of cells here, and they have dendrites, and they got spines. They're kind of like pyramidal cells. But if you look at the inputs, what happens, the turtle has a dorsolateral geniculate nucleus, and that comes up, and it just sort of projects across, it synapses on these cells, but there's not very many cells up there, and that's kind of like the original layer 1, mostly dendrites with just some axons coming up and synapsing on cells. That still happens. There's still input that goes to layer 1 from the geniculate even, and a lot of dendrites up there. So that's the original input layer. But because mammals evolved the subplate and maps and all this sort of cortical lamination, most of the action is sort of going on in these other areas. And so what are these other areas? Well, the next one we'll put in is layer 4. And so layer 4 is the input layer. So in general, input coming into a cortical area is going to tend to go to layer 4. Now there's a lot of input to the other ones. We'll see how that works out, but you can sort of think of that as the input layer. And so if we look, for instance, at the dorsolateral geniculate, lateral geniculate nucleus, what does that do? Well, it comes into layer 4. That's one of the major inputs. So then we have layer 2, 3. And people initially sort of distinguish 2 and 3, and they're slightly different, but we can think of this area as the feed-forward, layers as the feed-forward layers. So these are sending information to higher level areas. So if we go to a higher level area, so let's draw a higher level area over here. You can see, as you might expect, that the output coming out of layer 2, 3 is going to go to layer 4 of the higher area. So here's 4 of the higher area. So what you'll see is that you got a neuron in the feed-forward layers, and that is going to go and provide input to layer 4 of the the higher cortical area. Higher cortical area. Now you might wonder, well, how does it get to layer 2, 3? So you've got to have some, you have some projections that project out of layer 4 within the cortical area, so that information can sort of get to the feed-forward layer and then go to layer 4 of the higher area. So let's go back here. We've got two more main layers. We have layer 5 and layer 6. And so layer 5 is basically the motor output layer. So for instance, if you go to motor cortex, that's where all the big neurons are that project onto the spinal cord. But you might wonder, what about V1? What's the motor output in V1? It turns out that layer 5 in V1 goes down to the superior colliculus, and the superior colliculus is a motor structure for moving the eyes around. But if you're a dog or a cat, it moves the ears around and the whiskers, and it moves the upper. In a frog, it moves like the upper body to rotate the frog. So every area has a motor output. So it's not like input comes in, you do some stuff, do some stuff, do some stuff, and you get motor output. It's not like that. Input comes in, immediately goes back to motor structures at every single level. So every single layer, every single cortical area has motor output. So motor output is being siphoned off all along. So there's a lot of other things besides just the superior colliculus. There's output to the basal ganglia, and we'll talk about that, like the caudate and putamen or the claustrum. There's output to the cerebellum that comes out of layer 5. So that's the motor output layer. And then layer 6, we can call the feedback layer. And so if you go to layer 6, you'll see neurons that go back down to the LGN. And actually sort of synapse cells in the LGN. And again, you've got to get some input into that layer, so there'll be some input coming out of layer 4, so that the feedback layer 6 has some information to work with. Now, if we go up to this higher cortical area, it's got a layer 1, and it's layer 2, 3. There's 1, 2, 3, 5, and 6. And then if we look at the feedback, what you clearly see is that the feedback comes and it basically just avoids layer 4. So it comes back and hits a whole bunch of different layers, layer 1, but it specifically avoids layer 4. And so because of this pattern, you can basically approximately order the layers, order the cortical areas by looking at like which layer the inputs and outputs go to. Now, it's not completely ironclad, and sometimes you have areas that look like they're at the same level. So you can imagine, you might imagine an area that has a feed-forward projection to layer 4 of one area, but then that area in return has a feed-forward projection to layer 4. That happens sometimes. So it suggests like those areas are approximately at the same level. They're kind of peers, as opposed to sort of progressing through a hierarchy. But the important point is that information from this higher level area gets back in big time to these earlier areas. So the lower cortical area has a mix of information from multiple different levels of processing in it. So don't think of it as really just a strictly kind of serial processing. There's some ordering to the processing, but clearly within 20 or 30 milliseconds, information can start coming back in from higher areas going back down to lower areas. And this is very important for how the brain works. So it's not a feed-forward system, despite using sort of feed-forward and feedback. You can put those in quotes. Okay, so that's our basic, and then all the same things that I drew here are happening over here. So there's output going to the caudate and say the putamen, cerebellum, superior colliculus. So there's all these motor-like outputs coming out, and then there's going to be feed-forward output that goes to layer 4 of an even higher area. Okay, so that's our sort of general scheme for cortical layers, and now we'll actually kind of look at what's going on in V1. So in some respects, looking at V1 is not a good thing. And why is that? So V1, visual area 1, or sometimes called striate cortex, is the most differentiated area of any area in the entire cortex. So it's the weirdest area of any area in the entire cortex. It's got the most cells in it. It's like got almost 3 times as many cells per unit volume. It's like super active compared to other lazy parts of the cortex. So in some respects, it's not that good to just concentrate on V1, but there's a ton of research on V1. And to be fair, we're visual animals, and the visual modality is by far the dominant modality in the cortex. And this is the first stage in the dominant modality. So, it's okay to study it, but just keep in mind, it's a bizarre area. It's weirder than any other area in the cortex. So when people first went into the cortex, like Hubel and Wiesel, to try to actually study how the cells respond there, so what were the early expectations? Early expectations. Well, if we go back to my very first lecture where I talked about this idea of the cell doctrine and in general, how does the cortex work? Stuff gets presented to your common sense, and then that starts going in and you do higher level things with it. So when people first went into the cortex, they kind of expected, there should be we should expect truth and beauty and complex, thought and things like that. So why were Hubel and Wiesel so influential? Well, what they did, to take up another theme, is they actually looked at the literature. And one of the things that was in the literature was this study by Letvin and Maturana. And that was in the frog. And they went into the superior colliculus of the frog and said, we found these cells, they're actually retinal afferents. So they're actually retinal axons coming into the colliculus, and they were recording from the retinal cells, but in the colliculus. And they called them bug detectors. Bug detectors is not a very good thing, because actually what they were little crawling worm detectors. And so it turns out if you make a little elongated thing and move it in the direction of its elongated dimension, the neuron really fires a lot. And so this notion that you've got sort of these lower level feature detectors in the cortex was actually sort of transported up to Hubel and Wiesel. And they were saying, what they did was basically, let's not think that the cortex is doing these super high level things like thought and truth and beauty. Let's just look for some understandable feature detectors. And that's actually how that's kind of like the historical sort of basis of the original Hubel and Wiesel studies of the cortex that discovered orientation, selectivity, and complex cells and simple cells. We'll talk about that next time a little bit. Okay, so what we want to do now is fill in the details on this side. So we'll make like a big sort of layer cake picture of what a little tiny chunk of V1 looks like. And sooner or later, I usually ask some question about this picture. I'm not asking you to reproduce. I'll never ask you to reproduce the whole picture. There's too many parts. But I might ask some question about it with some additional cue information in it to sort of jog your memory. So what we're looking at is just a small little chunk of V1. And we're specifically looking at a small little chunk of macaque monkey V1. Macaque monkey. It's very similar to human V1, not exactly the same, but similar. And so let's draw a nice big chunky piece. So this is just like a little piece, just a couple millimeters of V1. Okay, it's a little distorted. I don't like that. I'm gonna try one more time. Too tilted for me. Okay. Yeah, that's better. Okay. Okay, so that's a little cube, just a couple millimeters worth of V1. And so one of the problems, that you have with neuroscience is, people make additional progress. And so as they make additional progress, they sort of rename things. And so the original idea was that layer 4 was the input layer, and that's kind of like how it was defined. They said, we'll call it layer 4 if it gets a lot of input. But biology is always messing around, and so what happened? Well, some of the inputs went outside of layer 4. But people just had it in their mind that layer 4 should be the input layer, and so then they had to subdivide it. And so the way they subdivided it in macaque monkey V1 is they subdivided it into 4 layers. Okay, so and 2 of them are kind of like a subset. And so what are the damnable names of these things? So this layer is called 4A because it receives some input. This one was 4B. Turns out that doesn't receive any input. And probably what happened was some additional input kind of climbed out of layer 4 and went into the bottom of layer 2, 3. And then there's 2 layer 4C alpha and 4C beta. And we'll see that those are the 2 main layers that receive the parvocellular and magnocellular. It's kind of like the more sustained response, smaller receptive field, and then the more transient response that emphasizes changes. So those things are separated when you go into the cortex from the LGN. And so if you look at those inputs coming in, so there's some magnocellular inputs, and those guys go into layer 4C alpha. And then there's the parvo, so this is the Y-like input, the transient, more transient, larger cell. And this is the X-like input, that less transient, more indicating just how much of the stimulus is there. So this is sometimes called sustained. And so this guy projects into layer 4C beta underneath. Now, if you should ever go to the literature, which, who knows, might happen, and you look at something from cats, because there's been a lot of study of cats in addition to macaque monkeys. They don't have the layer 4A business. And so in cats, they call this guy 4A. I know, it's horrible. And they call this guy 4B. So 4C beta is called 4B in cats, just because they're missing that extra 4A input. So here's our other layers, like there's layer 1 up there, this is layer 2, 3, oops, let's put it in red. Layer 1, layer 2, 3 over here. And then we've got layer 5 and 6 down here. So that's our basic setup. This was known for quite a while. And so then how did it sort of get modified? So when Hubel and Wiesel were first studying the cortex, what they did is they would go into the cortex. So here's the cortex. Here's the layers in the cortex, and they would come in from the side like this, make a very fine electrode that's coming in like this, and then slowly move it horizontal to the cortex. And that's how they discovered these orientation columns. And some of their very early observations are very striking. So if you look at, the orientation here, you'll find as you move along the cortex, there's a particular orientation, and then suddenly there'll be a little jump, as if there was an actual sort of discrete column, like that. And then if you go a little bit further, there'll be another little jump in orientation. So there'll be multiple cells. And those things, those distances, so this, this is the distance. It's very small. We're talking, this is like, say 50 microns. That's equals 1/20th of a millimeter. So that was the initial Hubel and Wiesel observations. So that you go into the cortex and you move the electrode tiny bit by bit, and you see these change in orientation selectivity. So then what happened? Well, some enterprising people were studying the cortex, and they just went and stained the cortex with cytochrome oxidase. Now, stain the cortex for cytochrome oxidase. So what is cytochrome oxidase? So cytochrome oxidase is just one of the major enzymes in the energy processing, ATP generating parts of the cell. So every cell has cytochrome oxidase. So it's not specific to certain cells. It's just that there's different amounts in different cells. And so in the 80s, Margaret Wong Riley stained the cortex for cytochrome oxidase, and she found these spots all over, little spots all over the cortex. And so she sent the slides to David Hubel, and said, what's this? How does that fit into your scheme? And he looked at the spots, and he said, wow, I don't know what those spots are. So he put the slides in the desk drawer, but he got back to it. To his credit, he got back to it and actually started trying to find those spots and see what was going on. And what it turned out was the cells there were not orientation selective. That's a big shock. So the cells that had a lot of cytochrome oxidase turned out to be not orientation selective at all. And so this is a good example of, like when you're reading the scientific literature, you always got to take it with a little bit of a grain of salt. So the original story was everybody is orientation selective. How did they get that wrong? Well, one of the things that they did was, if you're recording with an electrode, if you damage a neuron a little bit, say tear a small hole in it, what happens? It'll get depolarized. So just think back to the very first lecture. You've got the membrane and you got the resting potential. The resting potential requires a semi-permeable membrane that just lets potassium out in order to make the inside negative. But if you tear a hole, well, sodium and potassium are going to all go both ways and make the cell depolarized. It'll aim the cell for zero because now there's no longer any selectivity. And so whenever you damage a cell, if you're recording from a cell, you often hook it up to a speaker. And so as you move the electrode, you go deep, deep, move the electrode in tiny little bits. Every once in a while you hear, it's like you nailed a cell, tore a small hole in it, and then the cell got really depolarized, and then it died. You don't do that very often, but that happens. And so they thought, maybe these cells are just damaged, the ones that are not orientation selective. And so they kind of said, we won't count those guys because we probably hurt them when we were recording. But no, they weren't damaged. They're actually just not selective to orientation. So that turned out, so after years of research, people started looking at these things, which they eventually ended up calling the blobs. So here's this guy is a blob or a patch, cytochrome oxidase patch. So high, cytochrome oxidase. So it doesn't mean there's no cytochrome oxidase in between. There's plenty of cytochrome oxidase. Every cell has cytochrome oxidase in your entire body. So it's just higher there. So these blobs extended down to layer 4, like that. But then they didn't go through layer 4. So it turns out there's a ton of cytochrome oxidase in layer 4. So if you actually look at layer 4, especially 4C alpha, tons of cytochrome oxidase in layer 4. But then if you go below in layer 5 and 6, you can see the blobs pick up again. And so there's the blobs. So we've got these blobs in there. And so where does the input to the blobs come from? Well, it turns out the coniocellular layers actually go up and synapse outside of layer 4A. So they didn't find this at the beginning, otherwise they would have had to have extended layer 4 even more. But basically, there are some inputs that come from the coniocellular layers that go to the bottom of layer 2-3. So this is why you can see, like, you don't have to remember everything. There's too many parts here. But okay, so now but where does this business fit in? The orientation selective cells, because they were definitely there. And so it turns out if you go into the area in between called the interblobs, and then record, you'll see there are different orientations as you move across the cortex. So that's what these things are supposed to represent. What is the orientation that is preferred? And so those are sometimes called the orientation columns. So say we take this guy right here. So that you can imagine there's a little column that extends small. We're talking like a 20th of a millimeter. It doesn't go through layer 4. The cells in layer 4 are not orientation selective, but then it picks up below like this. So this little thing is an orientation column. So this is orientation column. We haven't defined what orientation is versus, say, direction, but we'll just, okay, so there's our orientation column. So it's a little thin column that extends through the cortex. And the cells there actually are orientation selective. So now what about layer 4B? So we had the input coming into to the bottom of layer 2-3 into 4C alpha, that's the magnocellular layer, and then 4C beta, which is just underneath it, and that's coming from the parvocellular. Layer 4B doesn't get any direct input, and so it turns out that cells in layer 4C beta and 4C alpha project up into layer 4B. And it turns out that layer 4B is really direction selective. And so what is the difference there? So if we're testing for orientation columns, so here's orientation selectivity. So what does it mean to test for orientation selectivity? So basically what you do is the story was that the original orientation selectivity was detected when the human weasel had pictures of, monkeys and other things they're showing to V1, and they realized when they put the slide in, the dark edge of the slide gave a huge response, and they said, maybe it's not the picture of the monkey, maybe it's actually just the edge. And so that was like, so how do you do that? Well, typically, so that was moving, and that sort of complicated the picture, but you can also just test it with a little flash. And so if we have these circles here, which we're going to define as the receptive field. So what is the receptive field? It just means the part of the visual world that causes a particular neuron that we're recording from to fire. So that's the definition. Sometimes it's just called RF, commonly called RF, the receptive field. So it just means like you've got a visual map, and so you've got to go to the exact right part. These receptive fields are very small. They might be, just like they might be about as big as the moon. So if you look up into the sky, the moon is about 1/2 degree of visual angle. So these receptive fields are tiny. They're just cover the moon or 1/2 of the moon or something like that. And so you've got to find it. It's like a wild goose chase, because the animal's anesthetized and looking straight forward with its eyes open, and then you're going around like, where is this thing? And then finally you find it, like right there. Just that little tiny spot there is what causes that cell to fire. And so what I've drawn here is that tiny spot, and then I've made 4 copies of it. They're all in the same place, but I just moved them off to the side. So now we can do an orientation selective test. And what would that be? Well, we could start with, say, a vertical orientation, just flash on a vertical orientation, and then flash on a 45, and then flash on a horizontal one, and then flash on kind of like a negative 45. So that's a test of orientation selectivity. It doesn't have direction in it. It's just just the position of a static edge. So direction selectivity is different. So direction selectivity, remember the idea that how do you do direction selectivity? It's going to be something like we've got a detector, and then we have a little bit of a delay, and then we add that to another detector that doesn't have a delay, that's in a slightly different place. So here's this is a little spatial offset, and then something like that. If you move an object in this direction, what happens is it stimulates this detector, gets delayed a little bit, buffered a little bit, and then a little bit later in time it stimulates this detector, and then those two add, and then you get a response. But if you go the opposite way, it doesn't work. So it doesn't work when you go this way. Because in that case, the two responses wouldn't get superimposed on top of each other in time. So how do you test for direction selectivity? You could use a bar, but you can also just use a little spot. So we could just use a little moving spot. But now there's a problem. There's more directions than orientations. So we got like twice as many directions as we have orientations. So let's just do it with a little spot. So what you could do is you could just, move the spot in that direction, and then move the spot in that direction, and then, move the spot up, and then move it in that direction. But now we've also got to test the opposite direction. So now we're opposite, and so you got to go through 360 degrees of testing in order to test. So here we're going like 0 to 180. Here we're going 0 degrees to 360. So that's conceptually different than orientation selectivity. And it turns out the cells in layer 4B are direction selective. They don't need an edge. They just, even a little moving spot is fine for them. So there's a whole layer that's sort of specialized for this direction selectivity. Now, you might have heard of yet more columns, like what about ocular dominance columns? So it turns out if you look at the input coming in to layer 4C alpha, it mainly comes from one eye. And so what that looks like is there'll be like a chunk coming from, say, the right eye. So let's put right. That's right eye. And then right alongside of it, there'll be another chunk that just comes from the left eye. And it's representing the same part of the visual world, but from the left eye. And you can see, wow, this is starting to get really complicated, because how do you deal with the map? Because in order for a cell like that in a higher area to compare something between the left and the right eye, it's going to have to, the axons are going to have to go up like this, and then they're going to have to sort of move sideways in order to sort of synapse on each other and actually do a comparison between the left and the right eye. So somehow, because you actually displace the two copies of the left and the right eye in the cortex, in the cortical map, because the cortical map is still there, and then we've got to deal with that. So you can see, like, it's just the complexity is just piling on. So those things are called ocular dominance columns, but they're not actually columns, unfortunately. So these are called ocular dominance. They're actually ocular dominance stripes. Sometimes they're called columns because when they first looked at them, they look sort of like a little column. But if you look at them from the surface, from surface, it's like if you just pull out layer 4C. So if you just pull out layer 4C like this, so here's 4C. It's got, like, the alpha and beta part, so 4C. What do these columns look like? They look like zebra stripes. They'll go along like this. And they might have like a branch in them occasionally. So there'll be like one eye like that, and then in between there'll be the other eye, and then there'll be another stripe over here, something like that. So they're actually like little zebra stripes. They're not like columns, like sort of the orientation columns, which is like a little tube. Okay, so what's the bottom line here? Well, the bottom line is, what we've done is we've taken this information that was coming in from these three main chunks down here. So if there's Magno, that's not direction selective or orientation selective at all. Parvo, that's not direction selective or orientation selective at all. Conio, same. None of these are direction selective or color selective, or direction selective or orientation selective. And so what we've done is we've combined information from them and constructed some higher level streams. So what are those higher level streams? So this is dom stripes. So what are those higher level streams? So there's three higher level streams. So the first one First one are the blobs. So what are the blobs actually doing? So they're not orientation selective. So they're surprisingly not orientation selective. So why would you want to sort of, if you've got all this orientation selectivity, why would you want to get rid of it? So what are the blobs? Well, they're just the donut holes. You know, so here's the donut hole in layer 2, 3, and then there's a gap through layer 4, and then here's the bottom part of the donut hole in layer 5, 6. So it's just the donut holes. These guys are not orientation selective. Hume and Weasel discovered that they were color selective, but it turns out if you look at animals that can't see color, like a bush baby, they still have blobs, or a cat. This cat still has blobs, can't see color very well. So it turns out, what's the original sort of like, the original purpose of the blob system? And it's basically for brightness. So what does that mean? Well, if you look at, we've got orientation, which tells us the orientation of an edge, but we might also just want to ignore orientation. An example of where you might want to ignore orientation is, imagine we have just a simple shape like a cylinder. And if that cylinder is being illuminated, so here's the sun, from that direction, what's going to happen? Well, what's going to happen is this part is going to be very dark, like that. And this whole thing is going to be slightly darker than the top edge, but it's going to be why don't I do it the other way? I'll leave it that way. Yeah. So one of the questions is I'm drawing with a marker that's light, so I want to reverse it. So light coming down from this side. So what does that do? Let me do it the other way. Okay, so light coming down from that side. What happens? Well, this is going to be light. And then as we go off to the edge, it'll be a little bit less light. Okay, and then this is going to be very light up here. So the whole top is going to be pretty light. And so what is the what's the point here? Well, it turns out that you can use information about the brightness. Like if you just look at my arm. So if you look at my arm, you can see like the underside of my arm is dark here, and then it gets brighter here because of where the lights are coming from. And if you look at each of my fingers, you can see there's a little dark thing underneath my finger, and there's a little bright part on top of each finger, and it's very dark in between these two fingers. You can use that information to determine the actual shape of an object. So, or if you look at my nose, you can see that it's lighter on this side because I'm closer to that light than it is on this side, and that gives you some information about the actual surface. But that information needs to know what the brightness is, and it actually needs to throw away the orientation. So say we had a little receptive field that was sitting over here. Well, that is going to have an orientation signal in it if it was orientation selective. And so what the cortex has done is it has destroyed the orientation signal. So, because all these cells around it are orientation selective in all around the blob. But what the blob does, it sort of combines all the signals and essentially blasts the orientation signal out of there and just says, I'm just concerned about the brightness of the object. And these cells have a very high dynamic range, which means they can signal different levels of brightness very well. So that's what the blobs do. And then what is color? Well, color is kind of like a multi-dimensional brightness. You got like red brightness and green brightness and blue brightness. But the original plan was just brightness, because plain old brightness is the way that you actually measure the shape of and perceive the shape of an object. So that's kind of like the general consensus. Now, if you go back to the interblobs actually want to destroy brightness. So these guys are orientation selective, but now so if you read, the problem when you read the literature is there's all this jargon, and so if you read the literature about orientation selective cells, what do they say? They don't say they destroy brightness. What they say is they have good contrast gain control. Good contrast gain control. So what does that mean? That means, like, you put in a high contrast thing, and you get an output. You put in a low contrast thing, you get about the same output. So what does this mean? So what this means in English is, ignores brightness. Because it doesn't really, you don't really care if you're trying to figure out the shape of an object based on its orientation. You don't care whether that's a bright edge or a dark edge. You just want to get the actual shape of it, and so you want to actually throw out the brightness signal. So this guy ignores brightness. This guy, ignores orientation. So this is how you can sort of see these streams sort of being constructed. Now, the last, so what are the interblobs? I always try to hopelessly draw this. The interblobs are like the Swiss cheese, right? So they're like, they've got sort of blob-shaped holes in them. Here's a little chunk of an interblob, and then there's, of course, another one for layer 5, 6 below it. There we go. So that's the little Swiss cheese. And then what about layer 4b? Well, layer 4b doesn't even go throughout the whole layers, but layer 4b is direction selective. So this guy is highly direction selective, and we'll have a lot more to say about this when we talk about how higher motion areas actually use this information coming out of layer 4b. But these cells tend to ignore color brightness. But we'll also see they kind of ignore orientation. It's a little more complicated than that, and when we talk about the aperture problem, we'll see how it works. But they basically ignore orientation, or they misinterpret orientation, we'll say, let's say. So these are the streams. These are the main streams, and then what happens? Well, they go up to higher areas, and then they get mixed together, and then we end up with some higher level streams. When we get to V2, so there's a lot of mixing occurring in V2, and then we end up with some higher level streams. So at the level of V1, so at the level of geniculate, the streams were the magnocellular, these transient ones and sustained ones, and then these conial ones that are blue. But after doing all this mess in V1, we have constructed 3 new streams that emphasize different things. So the blobs are more interested in brightness. They don't care about the orientation, and they're probably using this in order to sort of detect the shape, provide input to detect the shape of an object. The interblobs are interested in the orientation, but those guys actually don't care about the brightness. They kind of try to remove the brightness signal so that they just tell you where the edges are. And then layer 4B kind of ignores orientation and brightness and basically just tells you what direction something is moving. And just to really sort of hammer home the point in the last -1 minute, if you read the literature, what you'll see is people still have a feeling like, these guys, these Magno guys, should be motion guys because they really give a good response to a change. And so can we find a pathway that sort of comes up through layer 4C alpha, and then it goes to layer 4B, and then maybe this goes off to, this in fact projects to area MT, which was a motion area. So could that be like a motion pathway? And the most striking experiment is not very well known, partly because it kind of goes against this idea that the simple idea that there could be a simple stream. But this was an experiment that was done by Bill Merrigan, if you want to look it up, Merrigan. And so the idea was, here's the Magno system in the retina. That goes to the LGN, the Magno DLGN, and then that projects to layer 4C alpha, and then that projects to layer 4B, and then that goes to, say, area MT motion. So that would be the motion stream. And if you read the literature, you'll see this diagram which I just drew here. So what was this experiment? Well, this experiment was, don't do this at home, was to damage all the Magno cells in the retina. So here's the retina. So it turns out there's a chemical, and it can just damage the Magno cells and not the parvo cells. So here's the parvo cells. And so what if we just nuke all these guys? So what happens? Well, if you test the animal, it can no longer see very low contrast stimuli. And that's not surprising because these cells are very transient and they respond to low contrast. And so that was that was the original hypothesis. Well, we just nuked the motion stream. So now we play a low contrast moving stimulus, animal can't see it. But what happens if you play just a slightly higher contrast stimulus? So if the parvocellular cells can see, you need about 10% contrast. So they don't respond if you have very low contrast. I mean, like not much of a brightness difference between 2 parts. So you need 10% contrast is a little bit more contrast. Well, if you do that, no problem. The animal can see total motion. All the motion thresholds are normal. And so what that means is that there's a pathway that is somehow coming up to layer 4C beta, and then it's getting into this other system. And so this is a classic example that just shows you it's a key experiment, not very well known, partly because it kind of violates your expectations that you have a motion pathway. But this shows that this is not the motion pathway. What's going on is you've got some streams coming in, and then they get all mixed together, and then you make new streams. And the new streams are the blobs, the interblobs, and this layer 4B direction selective stream. But that direction selective stream is made out of 4C alpha and 4C beta. It just doesn't go straight through. So I always like to emphasize this experiment because if you do an experiment and you have a hypothesis, I think the world is such and such a way because everybody else thinks it's that way, and you get some data back that says the world is that way, then you say, okay, yeah. But if you do an experiment and it violates your expectations, like this experiment did, those are the most important experiments by far. And those are the ones that maybe the graduate students start crying because, like, wow, it really didn't work out the way I wanted it to. Those are the most important experiments because they're actually really telling you something about the world, about how the world is. So when the experiment fails, sometimes that's actually your advisor will know that's actually sort of like a good thing because you found out something new. So this is a great example of that. They were totally expecting to completely blow out the motion selectivity when they did this experiment. And then all they did was turn the contrast up and they said, dang, they can see motion just fine. All the thresholds are normal. Okay, so I think we'll end the recording there, 10 minutes late, and ask for any questions.